All right, thanks very much to the program committee for inviting me to speak. I realize that we've had a long day, so I'm going to try to be animated, try to wake everybody up. I'm going to be talking to you today about a project I've been working on for the last 3 years called OpenWorm. My background originally begins in the world of computer science and artificial intelligence, professional software engineering. How are we doing?
Is your presentation all green or is there other color?
No, yes, green is the color.
Okay.
Yes, green background. It's green. There should be, yes. We'll see when we get to the movies, but I think it's supposed to be green. It's green on my screen. It's green and white. We'll see. Okay. Background in computer science, AI, software engineering, then took a detour into neuroscience. Spent the last several years as a graduate student in neuroscience, finished up with my PhD in that, focused on the intersection between computer science, computer engineering, and the details of the biology of the nervous system. And this project is to some extent an extension of some of my thinking there. So we're building a simulation of C. elegans. Hey, so we just heard about that, and that last talk was awesome. And so you kind of got some of the justification of why I focus on small animals. I'll talk a little bit more about that. I'm really interested to see more about that work. It's actually the first time I've heard of it. So OpenWorm is open science, which I'll tell you what that means. It is focused on computational neurobiology and software engineering. And so that'll be the second half of what I talk about. And the third part of my talk, if I have time, I'll say a little bit about the scientific approach that we're taking. So our goal in the long term is to build a full-scale simulation of a C. elegans. Okay, this is a project that we did not come up with ourselves. It was originally proposed by a computer scientist named David Harrell back in the 90s. And he sort of posed this as a grand challenge for computer science to be able to simulate a C. elegans. So we sort of pick up where David Harrell leaves off. But in the medium term, we have a bit more conservative goal on the way to get there, which is to accurately predict a database of worm behavior using a three-dimensional neuromechanical model. So my talk's going to be explaining what that means, how we might do that. Okay, so a little bit about the project. So we're an international open science community. We're started in 2011. There's 9 core members, meaning 9 individuals that meet on a regular basis to work on the project. We've amassed 36 contributors across 12 different countries. We just gained the 12th country last week. Actually, a scientist from Iran is collaborating with us and contributed some code to us. Very exciting. We have 24 repositories on GitHub and 220 followers on GitHub. We have 300 folks on our mailing list, so very active community. Our community builds everything in the open source. So essentially, we publish everything to the open source in advance of publishing it in academic journals. We put the code that way. We write our manuscripts in a collaboration, so that part doesn't go out. But the code goes out on GitHub in advance of everything, and that's part of the idea of the open science approach. Last year, we hosted more than 40 online meetings, principally using YouTube and Google Hangout. Okay? So this version of open science is talked about in the book Reinventing Discovery by Michael Nielsen. Okay? It's a good place to get a handle on what open science is, and we kind of use it a little bit as a guide for how we do what we do. Here's a list of our core team and our contributors that have been amassing over time. And before I get into some of the details of the how we do the simulation and the why, I just wanted to show you an example of open science at work, because sometimes folks don't really understand what that means. Okay? So we literally post our roadmap for what we're going to do, as an open science project does, as issues. Like this is an example of literally an issue that we posted up on GitHub using GitHub's issues mechanism. And we said, we want to create a Hive Plot from the connectome data that the C. elegans has. And we had some description about what a Hive Plot was. It's a particular way to visualize a graph. I'll show you an example in a second. And we said, hey, we have this data, this historical data from John White back in 1985, which is the complete connectome of the C. elegans worm, and we would like to combine these two technologies together. Here's where you can go get them. Here's what some code might look like if you went to go do that. Would anybody like to volunteer? And as people do, they find out about us via social media. They find out about us in a lot of ways online. We got an email from this gentleman Pedro Tabakov, who lives in Brazil. And he said, I really like the idea of this project. I have this background. I would like to help. Here's his GitHub account. Okay, and we said, hey, well, you had this starter project, this Hive Plot connectome thing. Maybe this is something that you could work on given your background. And 3 weeks later, he made a commit to our public repository, created a folder with a Hive Plot representation in it. And what did that look like? It looked like this. So this graph, which those of you who are not familiar with Hive Plots, it's sort of a fairly new visualization technique. I actually really encourage you to check them out in and of themselves. I do another talk all about like Hive Plots. But what's valuable about them is that you can lay out all neurons in a graph on the same kind of axis. So it gets you away from the standard problem of hairballs, visualizing complex graphs. And so essentially you can lay out all the nodes of sensory neurons along one axis. I'm not sure if this is going to show up. Along the lower left axis, sensory neurons, the lower right, motor neurons, and then interneurons on the middle. And all those little edges that you see are the connections between them. And you can look at it for chemical synapses or gap junctions. You can look at all of them. This, by the way, also brings up a point. C. elegans connectome is the only whole animal connectome that has been produced to date. And it exists. And that then also poses the question of what are we doing with it? And I will talk more about that. But this is an example from beginning to end when I talk about open science. It sort of puts a coda on the open science discussion. Is that open science is about putting your roadmap up for volunteer inspection, recruiting volunteers to help you, and having those volunteers put something out. And we then credit those volunteers with doing that work. And when we write up manuscripts, they also get authorships. And so all sorts of exciting stuff that happens in that manner. Okay, so open science transition to why. Why C. elegans? We had some good justifications already, so let me just say a little bit more about that. C. elegans was first promoted as a model organism for biology by Dr. Sidney Brenner, who is still alive. He's emeritus at the Salk Institute. This is back in the 50s. He picked the C. elegans for many great properties that it has. First of all, it has reasonably interesting behavior. You would not believe that that might be the case until you have a look at several of the movies, but it is in fact true. So it does things like it seeks out food, it seeks out mates, it avoids predators, it avoids toxins. It has social behaviors. You can even knock out the social behaviors. They'll either clump together or they'll not ever hang out next to each other. And there's some neural correlates of that. It was the first organism that had its full genome sequence, so it's been very important for a lot of genetic work. We know a lot about its cellular anatomy, so we know the hermaphrodite has exactly 302 neurons, exactly 95 body wall muscle cells, and so approximately 1,000 total cells. And even more excitingly, we know every cell division from fertilized egg to adult, and it happens to be an organism where with extremely well-conserved divisions of cells, so that it's that way every time. It's sort of exceptional, and it kind of required a biologist to look in deeply enough to pick this organism, select it out specifically because it had those properties, and that's what Sidney Brenner did. And as I mentioned, it's the only full organism connectome that's been completed to date. Okay. So I believe, and we believe at the project, that C. elegans represents a huge opportunity for computational biology. I'm going to talk to you a little bit about that. So one of the things that we aim to do is to close a loop that hasn't really been closed in modeling work before, okay, in I think the way that we really need in order to move forward in computational biology. So those of you who play around with neuronal models of various degrees realize that if we think of a standard 3-layer concept of a neural network, we activate sensory neurons, that would lead to activation of interneurons, and that would lead to activation of motor neurons, and of course there's feedback, right? And a lot of modeling happens basically just here. We work on the network, we do learning, we throw all sorts of algorithms at this problem. We do this at high degrees of detail and low degrees of detail. Well, what you can do if you want to go and then model a whole organism is that there's some other aspects of, well, what do those neurons go and do? And what's interesting is that as part of the connectome of the C. elegans, not only do you know what neurons are connected to each other, you also know what neurons are connected to what muscles. So you could include in your model the activation of the muscles. But then, if you're going to do that, muscles are obviously moving something. So they're moving a body. And so you can model the shape changes of the body. And then that body, of course, then goes and creates forces in an environment, and then the environment changes and has an impact back on the body, and then that has an impact back on your sensory neurons. And so this loop is a very exciting thing to me and to us in the project, because what it has the potential to do is to give us the ability to constrain the inputs and the outputs of a neural network to something that we can actually measure and test. And that is really exciting. So what data do we have? We heard about some gaps and limitations in data in the last talk, and it's true there are some gaps for sure, but let's talk a little bit about what they are and what do they look like. So one thing that we have in C. elegans that's a real gift is data about structure. That's a happy thing, right? So examples of data that we have about structure, okay, are the nervous system. So this is, and I'll show you more detail about this later, but so this is, that's it. That's the nervous system of the C. elegans that you just sort of flew through backwards, okay? And I'll show you in even more detail what that is. But we have the three-dimensional anatomy, cellular anatomy of those neurons. Now, looking at this, and you saw an image like this sort of earlier, looking at this, I think there's some things that become very clear that when you're looking at real biological neurons, shape matters, it's kind of complex, it's sort of tangled. I'll show you a way to explore it in a moment, but this is something that exists, that we have the point-to-point connections and we have the three-dimensional anatomy, and so one of the things that we've done in the project is to put those two things together in a computational representation. You can go, this is actually posted on a resource called Open Source Brain, which promotes the use of NeuroML, which is a nice standard for neuronal modeling, and I'll say a little bit more about that later too. Okay. So, so we have structure of neurons. We also have structure, by the way, of all the other cells. I just don't have time to show that to you right now. If you go to browser.openworm.org, you can actually look at all this right now today for free. You can slide down, peel away the skin, look at the body wall muscles, see how the neurons are in relationship to the rest of the cellular anatomy. So we've got some really, really good anatomy and really good understanding of the connectivity of the neurons. Now, data about dynamics. Okay, so there's frequent concerns about dynamics because of a problem doing standard electrophysiology on C. elegans is that it's got a pressurized body, and so when you go and perforate that pressurized body, it all comes spilling out. So that's not so helpful for the physiologist, you know, if they get very frustrated with their prep. There are actually some physiological studies that deal with that problem, so it is still possible to record from those neurons. They're very small, but you have to deal with the fact that now your worm is no longer solid. But what's exciting about what's happened in the last 5 to 10 years, which I think should cause us all to revisit the C. elegans, are some trends with regards to optical imaging. So I'm going to show you 3 examples. This is the snout of the C. elegans being imaged using calcium imaging. And here an odorant is being exposed to the snout. And what you're seeing in the color pattern is the calcium activity of the neuron in the snout as it's being exposed to that odorant, changing color based on how it's being activated. So the nice thing about C. elegans is it actually has a transparent body. You don't have to do anything to make its body visible. So you can shine light right through it. So if you put any kind of new technologies that fluoresce, you can easily image things in the C. elegans body. Here's another example. That, by the way, was from Srikanth Chalsani at the Salk Institute. This one is from Andrew Leifer at Princeton University. Here, they've got neurons that are lighting up, again, with calcium imaging that are freely moving, and they're, in fact, tracking the freely moving worm. And so what you're seeing is sort of, it looks like it's sort of shining and shimmering on the inside. That's actually specific 2 or 3 neurons that have been picked out for its activity pattern, and so you can see it as it goes. He's actually got another microscope that even gets a closer level of resolution, like equivalent to what I just showed you as well. So you can see how it moves, and you can see the neurons working in collaboration with that. And this, which is very recent work from Schrodel et al. In Nature Methods 2013, and we just did a public journal club on this a few months back, so you can get the details. This is actually imaging in the head of the C. elegans, the various activities of about 100 neurons just in the head of the C. elegans as it's sort of confined in this particular example, but different activity patterns as it's being exposed, I think, to different levels of oxygen. So while we had been in an era where it was very hard to record the physiology and the dynamics of neurons in C. elegans, that is coming. And studies like this are showing that it's increasingly possible with new optical techniques. And when we get to put voltage sensitive dyes in these neurons, which is just starting, it's going to get even better. So that's exciting. Now, when it comes to this part of the cycle about body shape changes, we actually have a very good resource, and that makes us really, really happy. So both structure in terms of how muscles make the body move, but we also have dynamics. And that brings me to this example, which is work by Yemeni et al. This was published in Nature Methods also this year, last year in 2013. And so this is the worm behavior database that I sort of briefly mentioned as one of our goals. So 10,000 movies of worms, okay, taken over time. Like painstaking work, right? This field is sort of, on the biological side, it's sort of heroic data collection efforts is kind of a mainstay of this effort. It took 12 years to do the connectome originally, 12 years of work. So anyways, this is 10,000 movies of worms. Okay, they're being tracked, so you can see that the camera is actually following the worm as it moves along, and you can see a sort of a colored outline over its body, and that's actually image processing that's tracking the worm as it goes. It's reconstructing, it's segmenting out the skeleton of the worm, and then it's taking that skeleton and it's recording frame by frame the position and the posture of the worm over time. So to go even further, this wasn't just worms that have normal genomes. They also did this for 300 of the genetic variants, and some of those variants have motion artifacts. Some of those variants are known to not have certain neurons. They're known to not have certain neurotransmitters, and they move in a different way. Those were also included in this database. So what we're trying to do with the Open Worm project is that what we're currently working on right now, we're taking that data and turning that into basically a behavioral classifier. Okay, that should be something fairly familiar to folks here. That's basically a program where you can feed it an example of a worm moving, and it'll tell you whether that's accurate or not. It'll tell you how close that is to what it knows to be correct. Is it a worm or is it not a worm? Or is it more like a mutant worm perhaps? So with that, we have then the ability to do something pretty interesting, which is to take the output of a mechanical model of a worm, a mechanical simulation of a worm derived from the activity of a simulated nervous system, and ask it how close or far away is it from a real worm, okay, and get a quantification. And we think this is sort of the experimental ratchet that's going to help us improve the model over time and get closer and closer to sort of reality, to ground truth. Okay, so I think that's really exciting, and this is work that's being done by a couple of Open Worm contributors, Jim Hokinson and Michael Curie. Okay, so as I said, so just to kind of put the code on that, so the point here is to close this loop of activity, which doesn't happen very often in neuronal models, and it provides us a way to constrain the activity of a nervous system by sort of physical motion. So now I want to show you what that physical part is, because I haven't shown you much about that. So the story started here back in 2009 when collaborators who are now working on the Open Worm project, let me actually move this forward, in Novosibirsk published a couple of YouTube videos showing their efforts to build a simulated C. elegans in 3 dimensions that looks like this. And as you can see, it's kind of blocky and kind of chunky, but what you're seeing is the motion of simulated muscles that are pulling on this sort of wireframe. It's got simulated Newtonian mechanics and physics, actually, so the motion here is not CGI, it's actually the results of a simulation sort of crunching, and it's got these simplified neurons, which is all that text there that you see, all the VDLs, and those are names of neurons that are sort of activating to make this thing crawl around. So when the project started in 2011, those of us who had kind of collected around Open Worm found this video, and we reached out to the team in Novosibirsk and we started collaborating with them. And so this has now taken a few iterations. Separately from that, I'll come back to that physical part. Separately from that, as I showed you before, we've done work to collect what White et al. In 1985 knew about the connection patterns of neurons, and we've combined that with this 3-dimensional view that came out of Caltech and a project called Wormbase to build a 3-dimensional anatomy, leading to this browser, which is online. You can try it out, which lets you actually click on neuron by neuron and explore the neuronal circuit and see which neuron projects to which in its 3-dimensional neighborhood, which is pretty cool. You can also do this in 2 dimensions, and there are tools that do that as well. But what's nice about this is that it kind of gives you the perspective on how neurons are connected together in that nervous system. Okay, so a few months back, we upgraded that blocky model of the worm moving that you saw before. And the same team that's been working with us in Novosibirsk in Siberia have kind of upgraded to what I get to show you now, which is really cool. They call it cybernetic, based on Siberia, okay, which is kind of their cool thing. Their first thing was called cyber elegance, which I also thought was a really nice play on words. Okay, so this is the latest version of the model, and this is a 3-dimensional view as well. We're looking at it top down, but it is actually 3-dimensional with a fluid dynamics sort of particle-based model of the worm. And this involves an algorithm that's called smooth particle hydrodynamics that it comes out of computer graphics. The algorithm was built by these guys based on a dissertation, essentially. And the model inside this, what you're seeing sort of on the edges yellow and red colors that are changing on the outside. This is the equivalent of those muscles that were becoming red in what you saw before, kind of going on and off. And this is still a prototype, and this is still essentially a very simple pattern of muscle activity. But what you're seeing is that it's sort of mock swimming in a little tube environment. So it's what it's pushing there in the blue is a very thin film of liquid, okay, away from it. And so what you have now is this is actually elastic. So the same way that a worm is squishy and elastic and you sort of pull on it, it's rubbery, we've built a environment in which those physical properties can now be represented. And details of muscle can be built in, how stretchy is the muscle, how much force does the muscle put on the body wall. It has that property that I mentioned to you before, that it is pressurized. So if you were to perforate this model, it would also sort of deflate in the same way that real C. elegans do. So it's going beyond the simple Newtonian mechanics model that we had before into something that's now got the potential to be much, much more detailed and much more true to life. Okay, so the talk title is about aggregating simulations, and I wanted to say a couple things about our strategy for how we're putting these things together. Obviously, this gets into the topic of multiscale modeling, and that when you want to do that in a serious way, as several of our contributors do, you want to actually come up with sort of principles for how you might hook models together, and I know this is a topic of a lot of conversation and discussion, and so we have our approach. And so we wanted to build a software platform that would enable something like a neuronal simulator and something like that, that physical simulator that you just saw, that cybernetic simulator, to work together in a single environment. So we needed to bridge across different spatial scales and diverse models and algorithms, realizing that we would also need to sometimes bridge across different time scales, so that's another challenge. And so what's come out of that is another sort of a spin-off of OpenWorm, a sub-project that's actually being used by some nonprofits, some for-profits, and some academic institutions. It's called Geppetto. Okay, take a moment to let that sit in. The simulator is called Geppetto. Yes, it is a reference to Pinocchio. Okay. This was actually a crowdsourced name by the project, so we didn't make it up, but it was suggested by the community. So Geppetto has been the target for a serious simulation engine that can combine multiple algorithms together and combine data together. It's some things that are notable about it. It has a web-based front end, so it works through your browser, and it has a software stack that at the bottom of it starts with standardized data so that you can describe models in declarative ways. It takes NeuromL as one of its foundations. There's a standardized way to describe the physical model that I told you about as well. It breaks down different simulator components into modules that are based on the OSGI framework, so this is Java-based. And then it allows both an API front end and a web front end for folks to ask questions. So you can check that out at geppetto.org. Okay, so what's in progress? So the original work that I showed you with the blocky simulator is Palyanov et al. 2012. That's sort of the first publication that the project came out with. That's been out now for a couple years. This OpenWorm browser that shows the connectome has been available now for a couple years, and this spatial connectome in NeuromL has also been available. All this available on GitHub and also published. And so currently, what we're working on is to take what I just showed you with that cybernetic integration of that stretchy model and embed inside of it the nervous system model that we've got. And this means we're also doing more work to do much more serious inference around what we know about the ion channels of the neurons. There is a way to start making some inferences about what ion channels should be inside those neurons, and there's experimental technologies that are moving that forward as well. So we're working with folks on that. We're building that worm classifier that I mentioned to you as well, and then we're taking more steps to embody what you saw there as a video and put it into Geppetto so you can all play with it yourselves. So that movie of the worm moving around would be available in that way. And then I also wanted to mention, as we were here talking about neuromorphic engineering specifically, so a project came up about, I think, in the beginning of last year called elegans, or silicon elegans, Si as the chemical unit, elegans. And so this is funded by FP7 in Europe. It aims to reproduce C. elegans behavior using FPGA models, one per neuron is their approach. And here's where you can find out some more information about them. We're not directly sort of affiliated or related to them, but we've had several meetings with them, and I think they're doing really interesting work, and I think that it would certainly behoove anyone who's interested in this space to reach out and get in touch. We talk with them on our mailing list. We have regular conversations with those guys, so they're doing good stuff. And I also wanted to point as well to other work that's happened in this space. Let me just reveal all those. Yeah. So other work that we're building on as well comes from Netta Cohen at Leeds, lab of Corey Bargman and Larry Abbott at Columbia University, lab of Sean Lockery, University of Oregon, have all prominent names in addition to others that have worked on C. elegans modeling over time. And the Nemalode project, which is another one by a gentleman named David Dalrymple. Okay. So in the last few minutes that I have, I wanted to just say a few remarks on the scientific direction, scientific approach that we have, and just kind of make an analogy to why this approach is something that's interesting. We're focusing on a system with very small number of neurons, and why do we do that? And this thing probably doesn't have cognition, and all sorts of concerns that we get. So as we all know, the transistor is the fundamental information processing unit of the computer, right? But imagine that tomorrow we forgot everything that we knew about how to engineer computers, and suddenly a MacBook Air, landed from the heavens, and we were trying to figure out what it was, and we started unpacking it, and we found a hard drive, and we found a monitor, and we found a keyboard, and we found all these things, and when we started taking it apart, and then we started building these theories and models of how hard drives talk to monitors, and how keyboards, record things, and we realized that it had a memory and that kind of stuff, right? But if we didn't understand the fundamental principles of how a transistor works, that it's an electronic switch, the thing that's at the core of all this, we'd be in trouble, right? So we kind of think of this as like the neuron is a transistor of the brain, but we still aren't satisfied that we have a model of a neuron that predicts all known biophysical data, okay? And that might be a sort of strong statement. Folks are free to disagree, but what we think is what a model needs to do, right, is it needs to accurately represent reality, right? The first computers were built to accurately predict where artillery shells were going to land, and that was based on using, physics equations, force equals ma, and it was pretty important that the shells landed where they were supposed to land, right? But in current sort of computational neuroscience, we have very, very few models of neurons that we can say that the neuron in the brain is going to do exactly the same thing that the neuron model is going to do. And so what we sort of think about is, what do we really need in order to build models of neuronal processing, right? This is the constant debate. How much biological complexity do you need to build in? And I hear that 2 weeks ago there was more discussion about this, and it's a continual debate, right? Do we need all of the complexity? Do we need not enough complexity? What we know is we want to build models that are accurate. We know that the standard is that if we fire the shell, we want it to land exactly where we predicted it to be. That should be sort of our reigning target. So how much do we need? So that's what I largely think about when folks say, well, they come up with reasons why not to do C. elegans, right? So we hear, okay, so sure, it has neurons, but do those neurons have action potentials, right? And we hear no. Okay, so it doesn't have action potentials. So it's a much simpler thing. It really has no relevance to what we know. But it turns out that these neurons still operate using the same biophysical processes, okay? And it's just really important. I just want to make this point really quickly so that that's all clear to everybody here, right? So Hodgkin and Huxley, back in 1952, demonstrated that the major feature of what neurons do, which is change their membrane potential, was governed by ion channels, right? Like that's the basic thing that they gave us. And so they showed us that if you stick an electrode in a neuron and you see how it's changing electrically over time, right? And you see that there's this change, this is the thing that we know is the spike, right? That's actually governed by sodium channels opening and closing and potassium channels opening and closing, right? They got the Nobel Prize in 1963 for this work. Okay, it describes how action potentials are initiated and propagated. And what they also gave us, which was really important, was a system of nonlinear ordinary differential equations that explain it. And that lets us model the membrane of the cell as a circuit. Okay, so many of you know this Hodgkin-Huxley type modeling, but fewer of you, I think, embrace it wholeheartedly in the way that we kind of do in the project. When you only have 302 neurons, you have the luxury of exploring these more complex models, right? So what we kind of think in the project that's really valuable that I just kind of want to leave you with is to think that perhaps the functional atom of a neuronal signal is not just a spike, but it's a change in the membrane potential. I think that's really what Hodgkin-Huxley showed us, is that you have to look below the spike in order to see what the real signal is. And I'll back that up in a minute. The other part is that the structural atom of a neuron is not just a node in a graph, it's not just a place where axons project to and dendrites receive, but it's an ion channel. It's the sodium channels opening and closing that make up that signal. It's the potassium channels opening and closing that make up that signal. And you see this when you look deeply into Hodgkin-Huxley-based models, right? For those of you that are not neuroscientists, these names, these words may not be all that meaningful to you, but neuroscientists again and again find these really weird things that happen with neuronal signaling that are really hard to explain if you don't factor in ion channel dynamics. So things like backpropagating action potential. These are things you can represent without doing Hodgkin-Huxley-based models. Don't get me wrong, you can sort of hack them in, but they come very naturally with an ion channel-based representation. So if we had that, if we had accurate biophysical models of neurons, some that would respect that we would run them and they would tell us exactly what the neuron is going to do in time, then later we could get rid of those details and use something simpler that's based on sort of the principles of what we want to let go, rather than what we see a lot, which is often sort of pragmatic concerns like it's too computationally expensive, or it's just the math is easier, or that kind of stuff. So what I leave you with in my last comment is that some of the trade-offs in neuromorphic computing can be that we can either make a lot of partial models of very complex neuronal systems that don't let us constrain them with real data, right? Or we can try to build holistic models of very simple neuronal systems that are very humble and may not have all the features of the exciting systems like Cortex, but that are perhaps that give us a lot of data to work with and that let us get as detailed as we want. So with that, just to respond to the charge directly, why would one invest in neuro-inspired neuromorphic computing over other alternative computing technologies? I think obviously neurons embody efficient computing architectures, and we can learn a lot from biological information processing of neurons because they've evolved for billions of years. The big wins would be, I think, building neuronal models that do what physics models do and accurately predict what real neurons are up to. How long will it take to get there? I don't know. And what are the critical path objectives? I think we still need better data, but we also need more detailed modeling, and we need more validation of detailed models against real data. Okay, that's my summary. This is my contact info, and thank you.
So some really, really cool stuff there. I wanted to take issue respectfully with one thing that you contended. It turns out that cortical cells can be very well modeled. Now they've had the advantage of being recorded from, and so they're models that are constrained by measurement. But you can build very predictive compartment model level models of cortical neurons, other vertebrate neurons. The real pity is that for C. elegans, and anyone that works on neural circuitry is sooner or later told, how can you figure out anything if they can't wire up the 302 neurons and figure out what the C. elegans does? Now you're cracking that problem. But of course the great weakness is that those darn cells are not modeled. And I guess one big problem is because you can't record from them. Now you showed that optical, these recent initiatives. Is there any, I mean, those are fairly gross measures. I guess the voltage die would take you beyond that to local changes in potential.
Yeah.
But you probably can then successfully model those invertebrate neurons just like we do successfully model cortical neurons today.
Right. In addition to the optical techniques, there's also been recent advances in culturing C. elegans neurons in a dish, which means that you can explore, you can pull patches, and you can explore the dynamics of the ion channels that a given neuron comes up with. And there's also, like I said, this sort of very painstaking work where you can let the worm sort of explode and record from some of its neurons as well. But yeah, so right, it has been harder to address, and that's why folks haven't done it as much. But I think that there is a lot of potential in the future for that. Yeah. So thank you.
So I like the story of the MacBook falling from heaven, and where you say rather than looking to the disks and the keyboards, we should rather find the transistors and understand how they work. But of course that argument also goes in a wrong way, because you know a transistor has many, many, many features that you absolutely have to ignore if you want to understand the principles of computation, like the analog behavior, the band gap structure, the rotation profiles and things like that. So you may end up searching for the exact rotation profile of the transistor or the analog features, and at the end you never understand what the underlying principle is. So what I'm saying is you have to know where to stop, of course, and you have to see what are the relevant features, the relevant degree of complexity that is important in order to understand computation. And that is a very hard thing. I mean, I have no recipe to do that, but this is what we have to do. And what I think one important way is, in a way what you propose, is that you go as complex as you can, and then you start in a controlled way, reduce complexity, switch off certain features, and see at which point the computational property or the behavior of the worm, for example, disappears. Yeah? So it makes no sense to go as detailed as you can, and at some point it may not be used for computation at all, but the question is not answered, of course. Right.
I like the comment. I guess, I don't know that there was one specific question from that, but you know, my reaction is that the luxury that we have with the transistor is that we built it, we engineered it, so we knew principally what to throw away, and we haven't built neurons, and so we haven't had as principled of a way to know what to throw away. I think that's right in line with what you're saying. Yeah. We have time for one more? Or?
Yeah, just a quick comment. It seems to me that the biggest limitation of simple organisms with identifiable neurons and so forth is that you're very much away from this paradigm where all neurons are essentially doing the same thing, and what you have are very specialized neurons that have accumulated a lot of specialized function rather than implementing specialized functions with generic neurons.
Yes. So as it turns out, there are a lot of different kinds of, there's a lot of diversity in neurons, and as much as we like to think that there is a great degree of uniformity in neurons that we find in sort of mammalian systems, and there surely is percentage-wise, because when you're talking of billions of neurons, you're going to have like any individual neuron may be more likely to be like some other neuron that's there, you still have a tremendous amount of diversity as well. So you've got dopaminergic neurons in the central nervous system. You've got neurons that are based on acetylcholine. You've got large neurons. You've got neurons that spike by themselves. You've got neurons that only spike when driven. So, and even in the cortex, obviously, we've got excitatory and inhibitory neurons. I think we're up to like 15 different types of inhibitory neurons in cortex. So, so there is specialization, but I think that, again, that's something that we may have to explore and deal with, you know, in some of these simpler systems before we can go back maybe to the more, to the larger ones. I don't mean to suggest, by the way, that any work that's happening with larger scale systems is not valuable. I don't mean to suggest that at all. That's certainly good stuff too. I'm just sort of suggesting that it's also good to explore the atoms and explore kind of what's down at the small scale. So I think my time is done. Yes? Thank you.